Assalamualaikum and hey guys, welcome back to our channel. In this video, we're gonna explain on thyroid cancer. With tagline, hashtag check your neck, boom! Introducing group mates consist of Harith, Jasmine, Nick, Faris, Najihah, Alia, and me, Shafika. Subtopics to be covered as below. Without further ado, let's begin.
Assalamualaikum warahmatullahi wabarakatuh. My name is Nurul Jasmine, and today me and my groupmate will be explaining on thyroid cancer. So cancer starts when cells start to grow out of control, and thyroid cancer is a type of cancer that starts in thyroid gland. So thyroid gland shaped like a butterfly with two lobes and is connected by a narrow gland called isthmus. So thyroid gland also produces hormones which are T3 and T4. So the main type of cells in thyroid glands are follicular cells and parafollicular cells. Parafollicular cells also known as C cells. Follicular cells uses iodine to produce thyroid hormones. So when a person is having too much of thyroid hormone, she might suffer from hyperthyroidism that can cause a person to have a fast or irregular heartbeat. And when a person is having too little of thyroid hormone, she might suffer from hypothyroidism that can cause her to slow walk or tiredness. And for C cells, the cells produces calcitonin, a type of hormone that helps the body to use calcium. And types of thyroid cancer, there are follicular thyroid cell derived tumor, which are papillary thyroid cancer, follicular thyroid cancer, poorly differentiated thyroid cancer, and anaplastic thyroid cancer. And for parafollicular C cell derived tumor, there is medullary thyroid cancer. However, it accounts just for a small proportion of thyroid malignancy.
Next is the risk factor of thyroid cancer. First is gender and age. It has been reported that thyroid cancer has 2.9 times higher rate in women compared to men. With an increasing age, the risk will also increase, in which will peaks at 40 to 49 years for women and at 60 to 69 years for men. Radiation exposure also has been major risk factor for thyroid cancer. It is noted that thyroid gland is more easily irradiated due to its position in the body and tendency to absorb iodine. Thyroid gland is characteristically radiosensitive during childhood and may persist over 50 years of life if acutely exposed at the young age. Ionizing radiation, such as X-ray, can cause DNA strand breaks and somatic mutations. Iodine is very important for synthesis of thyroid hormones and usually take in daily diet such as fish and dairy products. Conflicting result has been observed in epidemiology study where some studies showed high intake is associated with increased risk, meanwhile the other studies showed the opposite. In either way, iodine levels still can affect thyroid function and possibly occur in PTC via chronic stimulation of TSH and BRAF mutation. Studies in regard to obesity and TC showed that increased BMI will lead to increased risk of TC. There is also no established association between diabetes and TC. It was suggested that diagnosis may influence increased TC detection. Hashimoto's thyroiditis is a chronic autoimmune disease of thyroid which also can promote growth of TC. This can be caused by the elevated TSH level in hypothyroid patients with autoimmune thyroid disease as well as due to production of pro-inflammatory cytokines and oxidative stress. Lastly, lifestyle factors such as changing dietary intake, physical activity, and smoking may influence the risk of TC.
Molecular pathogenesis and mechanism of thyroid cancer.
Thyroid cancers can be majorly due to the genetic and epigenetic alterations of several signaling pathways. The altered signaling pathways involved in thyroid cancer are, firstly, is the MAPK signaling pathway. This pathway plays fundamental role in driving human cancer, such as maintaining human tumorigenesis, which is the cell proliferation. This pathway can be associated and cause PTC, papillary thyroid cancer. This altered pathway will first activate the mutations, including the BRAF V600E and RAS mutation. This actually can result in the expression of BRAF V600E mutant protein and causes constitutive activation of serine or threonine kinase. This is the figure that shows the classical MAPK pathway. This pathway can be started from an extracellular mitogenic stimulus that activates a receptor tyrosine kinase, RTK, in the cell membrane towards RAS, BRAF V600E, MEK, and ERK. ERK is activated by phosphorylation and enters the nucleus, where it upregulates tumor-promoting genes and downregulates tumor suppressor genes, as well as thyroid iodide handling genes, which finally lead to progression of thyroid cancer. The second altered signaling pathways involved in thyroid cancer is PI3K-AKT signaling pathway. This pathway was found and derived from Cowden syndrome due to the mutations of tumor suppressor gene called PTEN. This altered pathway can be associated and cause FTA and FTC thyroid cancers. The highly expression and activation of AKT can be observed in thyroid cancer cell. In PI3K-AKT, the extracellular signals due to the mutation can first activate receptor tyrosine kinases, RTK, in the cell membrane, which then leads to the activation of RAS and subsequent activation of PI3K. Activated PI3K catalyzes the conversion of PIP2 to PIP3. PIP3 activates PDK1, which consequently associates with AKT and leads to phosphorylation and activation of AKT by PDK1. The phosphorylated AKT, which is an activated form of AKT, enters the nucleus, where it induces tumor-promoting genes. In the cytoplasm, phospho-AKT also activates other signaling molecules or pathways. The most prominent one that we can observe here is the mTOR pathway, which has an important role in tumorigenesis by promoting translation. Phospho-AKT can also directly phosphorylate glycogen GSK3-beta and consequently inactivates it. GSK3-beta normally inhibits beta-catenin, however, it cannot perform the task properly. This consequently caused beta-catenin to enter the nucleus, where it promotes the expression of tumor-promoting genes. In the nucleus, phospho-AKT can phosphorylate FOXO3 on its AKT-specific motif. Such phosphorylated FOXO3 is translocated out of the nucleus to the cytoplasm, where it binds with 14-3-3 proteins to be sequestered in the cytoplasm, thus terminating the pro-apoptotic activities of the FOXO3 pathway. The downward arrow for the FOXO3 activities in the nucleus in the figure indicates this negative effect of AKT on pro-apoptotic genes in the FOXO3 pathway, which would otherwise be upregulated by the FOXO3 pathway. This unique coupling phospho-AKT to the three pathways provides a powerful driving force for thyroid tumorigenesis.
That's all from us. Thank you and have a nice day.